the term personalized medicine appeared, as far as I can tell, the first time it ever appeared was in 1971 in an article published in a Canadian medical journal entitled, "Can Personalized Medicine Survive?" Referring not to the way we think about it today, but to the demise of the family doctor who made house calls. So the answer was no there. And over the next 28 years, there was no mention of personalized medicine in the literature until the Wall Street Journal published an article in 1999 entitled "New Era of Personalized Medicine, Targeting Drugs for Each Unique Individual." And it was a highly influential article. It was, I think, stimulated by the combination of the Genome Project was well underway, and also by Wally Gilbert's bold prediction in which he said, in year 2020, you will be able to go into the drugstore, have your DNA sequence read in an hour or so, and given back to you on a compact disc so that you can analyze it. So I think his prediction has basically come true. And then in 2001, Francis Collins added fuel to the personalized medicine fire with his prediction, which went, by 2020, the impact of genetics on medicine will be widespread. New gene-based designer drugs will be introduced to the market for diabetes, hypertension, mental illness, and many other conditions. Well, we heard today, I think the many other conditions like cancer has come to be true, but I don't think there are any designer drugs I know for diabetes, hypertension, and mental illness, but our panel are experts on this, so we'll discuss that. But since the Wall Street Journal article and the Collins prediction, Google has recorded more than 2.4 million entries for personalized medicine. So personalized medicine, whether we like it or not, or whether we like the term or not, is here to stay. In fact, just last year, Weill Cornell New York Columbia Hospital created a new institute for personalized medicine, which they actually call precision medicine, which is now the new buzzword for personalized medicine. And then 2 weeks ago, the University of Pennsylvania appointed a vice dean for precision medicine. This is the first vice dean in this position, and also created a new institute. So despite this enormous enthusiasm for personalized medicine, its realization and reduction to practice faces four major problems, which will be touched on by our expert panelists, whom I'll introduce in a moment. There's complex biology, there's complex economics, there's complex bioinformatics, and there's complex physician-patient interactions. And I won't go into the details of those, but this will come through. And so is personalized medicine now ready to deliver on its promise, or is it decades away? And these will be questions that our panel will address. And the members of our panel, there's David Altshuler, who is a professor of medicine at the Harvard Medical School in the MGH, and also co-director of the Broad Institute. Richard Lifton, professor of genetics at Yale and medicine, and Huda Zoghbi, who is professor of pediatrics and heads a neuroscience institute at Baylor College of Medicine in Houston, and then Bert Vogelstein, whom you've already heard well introduced by Roy, and then Jonathan Sheldon, who is Director of Translational Medicine at Oracle. So what our plan will be is that each of our panelists, who are really experts in their area, will spend about 4 or 5 minutes giving you their views of the state of the art in their particular field, and then I'll ask them a few questions, and then let the rest of you then ask questions. And we probably have to move at a pretty rapid pace before the mayor comes in. But David, go ahead.
Thank you, Joe. So I was asked to spend 5 minutes talking about the state of the art of genetic diagnosis and personalized medicine. So I want to touch on 5 brief themes, or 5 themes briefly. Technology, comparison, evidence base, context, and biology. So those are my 5 themes. So technology. It's actually easy to forget that it was only 25 years ago that for the first time people could take a disease that existed in the human population and trace its inheritance to a specific gene and find the mutations in that gene. It's been only the last 5 or 10 years that it's been possible to do that same sort of approach for complex diseases where the problem is much more difficult. And it's only been a couple of years till you could sequence the genome of an individual and gather all that information. And so this is a remarkable advance, but it's certainly the case, someone commented earlier about the post-GWAS era. If what we're doing here is relating genomic variation to disease in its various forms, then we are at the very beginning to understand that. And it's very important to realize it's not that this is the end of some journey, but actually the beginning of creating that evidence base. And I think the analogy that we can all understand and we have to keep in mind is the beginnings of the internet and the World Wide Web, which made it possible to bring together information and make it useful. And it wasn't actually the first time someone sent packet switching information across the internet or made a web browser that you actually saw utility. It took many, many years to figure out how to gather, interpret, and make useful the information. Comparison. We don't know how to read genomes. We know how to write down genomes, right? We can now take someone's DNA and write down the genome, but reading it would mean being able to look at it and say, well, that gene encodes this function, or that change from a T to an A means you'll have this disease. And anyone who tells you they can do that today is simply trying to sell you something, because you can't do that yet. The only way we learn is through comparison. We compare the clinical characteristics of individuals in the population with their genomic variation and other measures, and we try and figure out those relationships. And so it is only going to be the case that we can interpret information if we can build an evidence base of high enough quality and large enough scale that we can learn these patterns. And when you realize that there are thousands of different diseases and there are 20,000 genes and each gene has many different variations, you realize that the scale of this problem is millions of genomes across all of medicine. It's not hundreds of genomes or thousands of genomes, it's millions of genomes. And so until we take seriously creating that evidence base, we're not going to be able to get as much out of this as we could. And I don't mean that there should be some monolithic effort to create that evidence base. I just mean the evidence base is needed. So I want to actually point back to the example of Vertex, which is a company I have the pleasure of serving on the board of, because if you think about that today, you can say, well, there's this mutation and there's a specific treatment and we should use it. But if you realize all that went into, again, this trajectory of finding the gene, of understanding the genotype-phenotype correlation, of understanding the cell biology, and then doing the drug discovery and the diagnostics and clinical trials, you understand that even in the best of circumstances, this is a challenging task, but we also see the remarkable advances that can come from doing it. And there's no one who was involved in that at all who imagines if you didn't have the genetic understanding that you would be in the position to treat and help patients with cystic fibrosis that exists today. Context. So I often get asked the question, and I think we might even be asked it from this group, should I have my genome sequenced, or how to interpret genome sequencing? And as in all things in life, context matters. So if a woman has a history, personal or family, of breast cancer, then a BRCA test has one interpretation. But if a newborn was studied for BRCA, or a person in the general population, it's a completely different scenario. We know in medicine, we talk a lot about prior probability of disease. And you think about when I was training in medicine and HIV testing, people knew even a test with 99% sensitivity and specificity, if applied to an at-risk population, had one interpretation, and if applied in the general population, had a different one. But for some reason, the reason, by the way, is genetic exceptionalism. This lesson is always lost on our field, where people leap from an evidence base created, let's say, in people with a disease to the general screening of the population. We'll probably talk more about that. And the last point I'll make is biology. There's the famous quote from the first Clinton campaign, it's the economy, stupid. I would say, to me, personalized medicine and DNA and genomics, it's all about the biology, stupid. It's the idea that if we can learn the biology, whether it applies to individual patients or populations, and do a better job of connecting what pathophysiology is actually going on in humans to what drugs we develop, we will do better. And that all of the rest of it, a little bit, is noise, as far as I'm concerned. The real question is, do we understand disease in patients, and do we have therapies that actually attack those underlying biological problems?
Okay, thank you, David. So let me ask you this question, which you hinted at, but you can expand on a little bit.
Sure.
I've read about healthy people who've had their genome characterized and are told they carry a mutation known to cause a severe early onset disease, and yet the person is healthy and doesn't have the disease. How is that possible?
Well, it's actually not only possible, it's inevitable and shouldn't surprise us at all. So there is this idea that because people studied rare Mendelian diseases, that all things that involve DNA are therefore deterministic and Mendelian, or even just because a collection of patients were studied with a rare genetic disease and found to have a strong enrichment for mutations, that everyone who has those mutations must have that disease. This is like saying we want to understand the relationship between smoking and lung cancer. So we go to lung cancer clinics where the people who have most severe lung cancer is, and we find that they tend to smoke more, a lot more than the general population. It does not stand to reason that everyone who smokes gets lung cancer. But because the technology for collecting DNA variation was up until recently very expensive and specialized, nobody was out sequencing the general population, and I'm not advocating that. But if you want to know, context matters, what it means to a healthy person with no symptoms to have a genetic variant, you have to have studied a lot of healthy people who don't have that to understand what the penetrance is. And we haven't done that as much. And so I think that it is inevitable that if you've only studied people with severe pediatric diseases, you will overestimate the effects, not because you did anything wrong, you did it for that reason, to make it more efficient, but it must be the case there will be people in the general population who don't have those diseases.
Okay. All right, Rick, tell us a little bit about cardiovascular.
Thanks. So first I'll start with David's theme of context. And in the broad context, where we are today is we know that there are about 21,000 protein coding genes in the human genome, and we know the consequence of mutation for about 3,000 of them. So I think from that starting point, we can agree that there's likely a lot of fundamental discovery left to be done. And secondly, that the technology that we have in hand today is really enabling, and there are no fundamental barriers to understanding the consequence of mutation of every gene in the human genome and appending that to human phenotypes. This is, I think, well exemplified in what has happened in cardiovascular disease as well as the other areas that we'll hear about. So as Bert so nicely demonstrated, cardiovascular disease, the rate has come down dramatically, and yet heart attack and stroke remain the number 1 and number 3 causes of death worldwide, which I think suggests that we want to make sure that we continue to develop new medicines and prevention strategies in this area. So if you look across causes of these major cardiovascular diseases, we have traditional risk factors, and we've been able to use genetic approaches to begin to reduce those traditional risk factors to molecular entities that afford new prospects for intervention. So for example, Joe and Mike's work on identifying LDL receptor mutations in patients with familial hypercholesterolemia, I think illustrate the fundamental point. The difference between using personalized medicine, personalized approaches to identify fundamental pathways and causes of disease may not be essential to then diagnose every patient with a specific cause, but you may find that the pathways that you identify from these rare patients lead to the identification of fundamental principles that can be broadly applied to the general population. And we certainly have that example with statins and reduction in LDL. So you go from rare patients to treatment of the general population. My own work in hypertension follows a similar paradigm, identifying extremely rare individuals in the population with extraordinarily high and extraordinarily low blood pressure, and identify the pathways that regulate how the kidney handles salt as underlying the overwhelming majority of these and are able to drive blood pressure across the entire spectrum seen in the human population. And within that, you can identify specific targets that appear to be very favorable for developing new approaches to the development of new antihypertensive therapies. As one goes on down the list and thinks about other areas in the cardiovascular system that we have not had prior access to ability to understand at the molecular level, there are whole ranges of diseases that are likely caused by sufficiently rare mutations, frequently de novo mutations that are absent in the parents, present in their affected offspring, but they so impair reproductive fitness that we rarely see the extended pedigrees that have been the mainstay of genetic approaches previously. And we're beginning to see this play out in a number of areas in neuroscience as well as in cardiovascular medicine, where congenital heart disease is now being found to commonly be caused by de novo mutations, and there are particular pathways that are coming out that were previously unrecognized as being involved in congenital heart disease, the H3K4 methylation pathway being one such pathway that has emerged from recent evidence. But the cardiovascular system is broadly very similar to all other areas of medicine, where if you look down the major entities, the targets that have been identified from fundamental genetic studies have transformed our understanding and the way we think about these diseases. If you think about cardiomyopathy or arrhythmias, there are rare inherited forms of these diseases that point to specific targets that may afford the opportunity for novel approaches to therapeutic intervention. And I think it's an open question as to how frequently personalized medicine approaches will be applicable to a range of these diseases.
Okay, well, thank you, Rick. So one thing, actually, Rick and Bert both touched on this. The decline in coronary disease in the U.S. in the last 6 years really is quite impressive. I think it's like a 60% reduction in myocardial infarctions. Yet, and if you extrapolate that line and nothing else happened, then coronary disease and MIs would disappear in about 30 years. But that's not going to be the case, because while this is coming down, obesity and type 2 diabetes are going up. And those are associated with the metabolic syndrome, for which coronary disease is a big part. So this curve over the next 20 years in coronary diseases will be very, very important in watching whether it starts to come back up. And that's a real bother. And then I think actually obesity may become the leading risk factor in cancer. What do you think, Bert? I mean, in terms of smoking was number one. You know, since obesity presumably affects, there are a lot of different theories. We don't have time to go into those while that's the case. But anyway, Rick, one question was whether or not you think the personalized medicine approach applies equally well to cardiovascular disease and oncology.
And I think, getting back to David's point, it all depends upon the biology. So if we look at cancer as a paradigm, we know that for every cancer, there are a modest number of mutations with relatively large effect on the risk of cancer. In all comers with cardiovascular disease, it is unlikely that we are going to have a small number of genes with very large effect in each individual. And this then motivates a somewhat different strategy for many patients, not all. But if you just think about LDL cholesterol and hypertension as predominant players in this field, it is much more likely that we are going to identify the key nodes in the right pathways and develop medicines based on those that will be applied to the general population. Then we are going to sequence each individual patient and give them a specific treatment to lower their LDL or lower their blood pressure. And of course, this remains to be played out, but there are new targets that have come out. PCSK9, as has been mentioned, is a beautiful example of this. And I think one of the challenges in cardiovascular disease is going to be being able to register new drugs on top of already existing very good drugs. And I'm concerned about the future in this area because when I started my internship, there was an iron lung in the corner. And of course, the iron lung was held over from the days of polio. And if we'd said, well, we've got pretty good treatment for polio, let's just stop there, we wouldn't have gotten very far. And I'm quite concerned that in areas like cardiovascular, where we now think we've got really good drugs in these areas, these are still the number 1 and number 3 causes of death worldwide. And we need to have strategies that are going to enable us to make further progress.
Okay, thank you, Rick. All right, Huda, will you tell us a little bit about neurodegenerative diseases and genetics?
So I'll talk generally about neurological diseases and touch on neurodegenerative diseases. And really what I'd like to do is sort of tell you how genetics have had an impact both in the clinic and in biology, and at the end touch a little bit about a path forward for potentially therapeutics. So neurology was probably one of the most dismal specialties to be a resident in about 20 to 25 years ago. because you couldn't do much for the patient. You couldn't even give them a diagnosis. What you could say, we think this might happen again, maybe 1 in 2 pregnancies or 1 in 4 pregnancies. That was the state of the art about 25 years ago. And if we fast forward to today, that really has dramatically changed. The most common Mendelian disorders in neurology, be it Huntington disease, many inherited ataxias, and several forms of intellectual disabilities and autism have been discovered, the genes identified, and you can diagnose these Fragile X, Rett syndrome, and so on and so forth. But what's even more fascinating and really only became possible through the recent large-scale studies in large number of patients looking at 2 areas of the, in diagnostics, copy number variations as well as medical sequencing. That's where I think the most startling findings came about. And I'll give you an example. If you to take all children with autism and intellectual disability, and if you were to sequence the most common causes of that, Fragile X, Rett, Angelman, and do screening for deletions, you're going to find the cause in about 20% of these children, because about 10 to 15% will have a deletion that's consequential that will cause a phenotype. But now you eliminate all these patients and you take a large group of patients who just walk into the clinic, they don't even have a recognizable syndrome, and they don't have any of those more common disorders, and you sequence their genome, their exome, you will discover a cause in about 25% of those kids. So when you actually tally, and I base this at least based on the recent publication in the New England Journal of Medicine from a large study from Baylor, in addition to many studies around the country, if you take it all together, 43% of children with intellectual disability and autism, today we have a genetic diagnosis, not an iffy one where the gene has been found time and time again. And I would say that thanks to the Simons Foundation, who really financed a lot of sequences so that we can know few culprits, and now we see them again, we recognize them. So I think that's really important, and that's only looking at coding sequences and big deletions. Imagine as we start getting better at computationally uncover regulatory mutations, non-coding RNA mutations, and so on and so forth. There is a big message here. A lot what happens to our brain, unless we fell off a cliff and hit our head, is really genetically determined. And that's something we should make note of and think about. So from that, let's move to biology. And what was the first thing we learned? When I worked on Rett syndrome, everybody discouraged me. That's a rare disorder. I'm wasting my time. I should work on a more common, important disorder like autism. Well, the big lesson we learned from this discovery is that there are no common disorders. Common disorders are a collection of rare genetic disorders. And I think this applies to autism, this applies to neurodegenerative diseases, this applies to cancer, and so on and so forth. I think the heterogeneity has been enormous. Several hundred genes for autism, tens of genes for Parkinson, ALS, and so on and so forth. The list will continue. 50 plus genes for ataxias. It's going to continue to grow. So I think that that's something we learned from that sequencing. So the families benefit from a diagnosis because they can relate to a group, and while there's no therapeutics immediately, being part of a group that can share symptoms and help what works and doesn't work has already been helpful. Of course, for adult Mendelian diseases, early diagnosis, pre-genetic screening, pre-implantation, genetic testing and screening, many families choose that, and it's been life changer for disorders with dominant inheritance. But the other, moving closer now to biology, what we're learning is these Mendelian disorders are really giving us insight about sporadic disorders. So I think you'll hear later this afternoon more about Alzheimer, but I'd like to just highlight the examples of Alzheimer and Parkinson disease. Less than 5% of Alzheimer is familial, yet we know it's APP mutations and it's presenilin 1 and presenilin 2, and at the end of the day, it's about the biology of a disease-driving protein, APP, that accumulate and or its product is a toxic entity in that disease, whether it's sporadic or whether it's familiar. So the biology is interrelated. The same in Parkinson's disease, although synuclein overexpression or mutation is a small percentage of Parkinson's disease, yet Most of pathology in Parkinson's disease involve the Lewy body that contains accumulation of synuclein. So that's one common pathway. The other area of biology is these additional Mendelian genes. If you take Parkin, for example, in Parkinson's disease, and PINK1, we're beginning to link them in a pathway, and we're beginning to understand that pathway impact mitochondrial function. So now we've got mitochondrial dynamics as a process in neurodegeneration. So this theme is going on, and whether it's lysosomal organelles, mitochondrial organelles, it's really coming from molecules to pathway, and it's the biology that's making the difference. And I know I don't have much time. I'd like to really move to therapeutics. How does genetics change the landscape for therapeutics? There are very neat, beautiful examples of personalized medicine. I'll give you one example. Patients presenting with dystonia. You treat them with dopamine because if you have reduction in tyrosine hydroxylase, you respond to dopamine. They don't respond. So there is something else. Comes exome sequencing and a discovery that they have a mutation in cypeptarin reductase, which is an enzyme critical for the synthesis of tetrahydrobiopterin. That's important for both dopamine and tryptophan. So if you add 5-hydroxytryptophan on top of the dopamine, you cure their symptoms. That's the dream way of personalized medicine. Sequencing changed the life of two people who were dysfunctional, having seizures, dystonia, to now they're functional and happy kids. Then comes the next level, where it's not so obvious, but years of biology and pathway analysis helped you. Take the situation of tuberous sclerosis and P10 deficiency, the phosphatase deficiency. Both can cause autism and intractable seizures. But really work from cancer and work on the PI3 kinase signaling pathway that we heard from Bert today help you put these two molecules in the mTOR pathway and bring up the possibility that perhaps rapamycin could be helping. And clinical trials are beginning to show that you can reduce the seizures from 300 a day to almost none to maybe a couple a day in some of these patients, and the data are still accumulating. So that's an example where really having the biology can help us as we begin to study some of these disorders. Of course, there's the area of do no harm. I'd like to bring the situation of presenilins and Alzheimer's disease. Gamma secretase inhibitors looked very attractive, but if we had really took the Notch data seriously, and we've looked at the decades of biology on Notch and how important it is, we would have immediately known, without even going through some of the clinical trials, how toxic these are. So the point I'm trying to make here, we've got ample discoveries, but the path to drugs is going to require both academia working on the biology, industry really working together reiteratively with academia to really cull out the best pathways possible. And two last points about therapeutics that we have to think about. Both of these disorders, whether childhood neurological disorders or adult degenerative diseases, the treatments will require decades. Therefore, they have to be safe. When somebody has to be put on something for 50 years because they have a mutation in APP or because they have a deficiency of an enzyme that causes seizures and autism and so on, it's got to be safe. And sometimes perhaps it's very hard to find a single target that you can inhibit and be safe. So maybe mild inhibition of multiple targets could be the answer. And I hope we would use human genetics again to help us point in the right direction. I would love to study, I was telling Joe yesterday, people who are octogenarians, centenarians, who have some genetic defects, but they're perfectly fine. Maybe they will give us clues about things like we can inhibit. So this is my overview.
Thank you, Huda. So a question that I'm sure you get all the time is, my 80-year-old father just developed Alzheimer's disease, and how can I find out if I'm likely to develop the disease?
So the Mendelian forms of Alzheimer, the genetically determined form that gives someone the risk of 50% chance recurrent, is usually earlier in the 60s and 50s. So the fact that this person and people, if there's no other family history, the odds is that this old gentleman has adult onset sporadic Alzheimer's disease. Age is the greatest risk factor, Anybody over 85 has a 50-50 chance of getting Alzheimer's disease. But of course, there's some additional risk factor. I think ApoE4 is probably one of the most important genetic risk factor. In the population, this genotype is about 15% in the general population, but in Alzheimer, it's about 40%. So having a single allele of ApoE4 immediately increased the risk to about 3 times. And of course, homozygosity makes it 12-fold. So it is most likely this is a sporadic illness. There is a slight increase in having a first-degree relative develop Alzheimer, but age is the biggest factor. And perhaps ApoE4 genotyping would be the second one that one can use.
Do you recommend that people that are curious have ApoE genotyping?
I personally don't recommend it because we don't understand the biology of how ApoE4 affects really A-beta clearance exactly and how we can go after it. There are no therapeutics today. It just perhaps will create anxiety. But if people would want to know and live with it, that's something that's a personal choice. But I think until there's treatment, I would not advocate that.
So the two famous genomes in genomics, arguably the two most famous, were released to the public. Craig Venter released his, and I think he's ApoE4
Positive.
Heterozygosity, and Jim Watson released his, but refused to know himself or tell anyone what his ApoE genotype was. So there's a lot of, even among the experts, about whether one should have testing. Bert, tell us a little bit more about cancer.
So I think the makeup of the panels today inform your question. The first 2 panels were composed mostly of people with strong ties to industry, and this panel is composed mostly of academicians. And this is important in light of what my son tells me. My son's an engineer, and he's always saying, "Dad, there's a big difference between interesting and useful." And our job as academics, in a sense, is to make this field useful enough that there'll be more interest in industry. Now, how do we do that? In oncology, it's probably the best example yet of personalized medicine in the sense that these companion diagnostics for EGFR blockade, for BRAF inhibitors, for ALK inhibitors are de rigueur. They provide the prototypical examples. So there's obviously been some progress, but this only applies to a very small fraction of patients. How do we get more patients to be usefully investigated through genomic medicine? And I would suggest 2 ways. First, in every phase 1 trial, and this was mentioned before, I think it's reasonable to start to include some sort of genome-wide sequencing of patients' tumors as well as their germline in the study. With the cost of sequencing continually going down, that really adds trivially to the total cost of the trial. And if that's done, you can get some surprises. Let me just tell you about one example, which I showed earlier. If that trial, initial trial of the PD-1 antibody, had done even limited genome sequencing of the colorectal cancer patients, they would have observed what we observed by hunch, that patient had a mismatch repair deficiency, and that could have started a whole new trial earlier, 2 years ago, on that subset of patients, and I think with positive and important results. The second implementation, and again, this involves collaboration between academia and industry. It's a perfect way to have that sort of collaboration, is to do more pharmacokinetics on patients in phase 1 trials and have that data correlated with genome-wide data to see whether there are any genomic determinants of differences in pharmacokinetics which could explain the great variations that patients have in the response to the same drug who have the same tumor. So I think there are very practical ways to make personalized medicine much more powerful and useful than it is today, and I hope some of those are undertaken in the future.
So all of us are curious to know whether you've had your genome sequenced. And if yes, why did you decide to do so? And if no, what would induce you to change your mind?
Well, I've had 3/4 of my genome sequenced, and I think when I tell you why, you'll understand what I think are good reasons to have one's genome sequenced. 3/4 in the following way. Several years ago, one of my brother's sons had a medulloblastoma, and we were interested in seeing whether there was any hereditary basis. So I sequenced him and his wife and his son with medulloblastoma, and because I sequenced him, that's half my genes, right? He inherited half of my genes. Unfortunately, last year, my younger brother himself developed a metastatic cancer, so I sequenced his somatic cell genome, his cancer genome, and his wild-type genome, and so that's the other 1/4, probabilistically. And I think that explains when I think genome sequencing is a valid and potentially useful enterprise at present.
Thank you. David, what do you agree or disagree on that?
Me?
Yeah.
I have not had my genome sequenced, although I've been offered it for marketing purposes by companies many times. And I basically think I agree with Bert. I don't have a question I want to ask of it, but I also agree with what Huda said, which is I believe it's a free country, and that if somebody else wants to have it done, we shouldn't demonize them. We also shouldn't be naive about the healthcare. I don't mean not the neurosis, but the downstream healthcare consequences of medicalizing everyone, something my Harvard medical students who I teach seem to get, even though some of the faculty don't, that if you give everybody increased risk of something, and then the primary care doctor is supposed to say, well, I have a variant of uncertain significance in BRCA. No one knows what it means in the context of a healthy person without any history, but I would like much more frequent intervention and screening. What doctors in our society are going to feel comfortable saying no? And so I think it's not the neurosis I worry about, it's the medicalization.
Okay, and Jonathan will tell us about the bottlenecks in bioinformatics and some of the ways that one can improve translation.
So I've got a few topics I wanted to cover and kind of give my perspective on where I think we're doing well and where we could do better. So, I mean, I think everybody, particularly this audience, is very familiar with the size of data we're talking and the challenges around storage. The European Bioinformatics Institute generates something like 14 petabytes every 9 months, and that's doubling at quite a rate. And I think Bert spoke about it very well this morning. I mean, we do genomics because actually we're pretty good at it. The reality is I think we all believe that it will be just one of a whole series of different tests that we carry out over the next 5 or 10 years as we get better at proteomics and metabolomics and lipidomics. The other aspect of the storage is really it's no longer about the data that I can generate in my organization. It's analyzing that data in the context of all the other data that's out there in the public domain. So even if you don't generate huge amounts of data yourself in your own organization, the reality is you'll be wanting to analyze that data with a much larger data set. There's the processing power aspects of the data once you've got over the storage hurdles. I mean, we're doing pretty good there. Alignments, 1 or 2 hours. De novo assembly of sequences, 1 or 2 days. I think an area that's often overlooked is actually just the simple retrieval of that data. So we've been doing some work looking at querying 100,000 whole genome sequences. So before somebody asked me, we modeled that 100,000 whole genome sequences. We took the 1,000 genome data set and then simulated that based on a normal distribution of variants. We were able to get sort of query times of a couple of seconds. So the reality is that I think the processing and the analysis of this data, at least at that level, isn't a bottleneck. Clearly, as we move on to more advanced modeling techniques, then it's going to be different. I think as long as anybody's been in the bioinformatics field, we've been talking about the need for standards. We're doing a little better now, I think, in the omics area. Standards exist for some of the formats that are out there. Clearly, as we move more into proteomics and metabolomics and other fields, we'll need to develop the standards to do that. I would also make the point, actually, that the reality is it's very easy to get seduced by the power of the omics, but in the absence of the context that we spoke about earlier, so basically the clinical data, whether that comes from a clinical trial or it comes from clinical practice, it's clear that we need to kind of bring those two worlds together. And there's a lot of challenges around the integration of that data, having consistent terminologies and normalizing that data effectively to do these genotype, phenotype kinds of analysis. The two topics I specifically wanted to call out was the need for biologist-friendly software. So, you know, as a bioinformatician, I mean, like the community, we get attracted into doing the cutting-edge stuff, but the reality is there's a lot of value that can already be delivered in the algorithms that exist. We just need to make them accessible to people. And that's a different set of skills, frankly, than a lot of the innovation that happens for the algorithms. You just need to put a decent user interface on them, enable, you know, cell biologists, biochemists, molecular biologists to actually work with that data. It shouldn't be the case that even a simple cohort identification task that means querying across genotype and phenotype, that shouldn't be the job of a bioinformatician. That is something that a scientist should be able to do in a self-sufficient manner. And that's so important because of the nature of the sort of iterative scientific hypothesis generation and testing that's so critical to science. So unless you enable the scientists to do that step themselves, the bioinformaticians are always going to be the bottleneck, and that's going to hold the field back. The last point I just wanted to make was just in the translation piece. So, I mean, we're doing a good job at gathering research data. It's telling us stuff without doubt. But if we want to translate that and get it to a point where it's clinical grade, i.e. where a molecular pathologist, a medical geneticist can say something of value about that data, that's a much tougher nut to crack. And there seems to be kind of two models that we're going down at the moment. And one is, they're very sort of polar opposites. One is around manual curation, which basically involves MDs, PhDs stuck in a room, trawling the literature, trying to extract information, assuming that it's been published in the first place. And there are clearly issues with that around scalability, in terms of that kind of approach. And then the other approach is a sort of global collaborative network, with the view that what's important will kind of rise to the top, and the quality of that will become apparent. But the reality is, in a molecular pathology setting, it's quite a risky business to say that you're going to let the good stuff kind of come to the top and you're going to make decisions based on that. So I would say, just as a general comment, it's that last piece that I feel personally needs some emphasis to really translate from research grade to clinical grade information.
Okay, well, at the end of the day, if the bioinformatics experts solve all those problems, let's hope they do, we're still left with, I think the real problems are that physicians are not trained in statistics, not to even mention genomics, and patients don't understand probabilities. So even if one has all this, I think that may be the really rate-limiting thing in translation and in personalized medicine, is informing physicians about statistics and them being able to explain probabilities to patients. But one thing, what should pharma be doing in genomics? What are your thoughts about that?
I mean, I think it's clear that a lot of the pharma are investing now in the biomarker discovery aspects of translational medicine. And I think it's very encouraging that the vast majority of clinical trials now in clinical development have a biomarker associated with them. So, the kind of infrastructure pieces are starting to come together. Actually, using that data in a meaningful way in the trial design, that's a different challenge, which we clearly have got a long way to go there.
So I think they're telling me that the mayor is out there, so we may have to cut questions short, but I did want to leave you all with one thought, that there's one true example of personalized medicine. It's probably the only example that I know of, real precision medicine, and that's your optometrist. and that's what one would like to have in all of medicine. But while the mayor's coming in, would anybody like to ask our panel any questions? Feel free.
One term I haven't heard yet is epigenetics. So, how do you play off the sequence versus the variants of expression of that sequence? I'm throwing out to the panelists to your nod your head rest. So.
Well, I will start, and maybe others can build on that. There are actually very few situations of an epimutation, so a mutation that would affect cytosine methylation, let's say, and may cause, in turn, in neurological disorders such as Angelman syndrome or cancer. But there are some additional ones that are yet to be discovered, and there are clearly a lot of epigenetics changes in the genome that might affect the level of key proteins that may be important either for cancer or neurological function. And those are, again, yet to be discovered.
But certainly we have the tools that we have today are we can sequence DNA very rapidly, very inexpensively. A major barrier in humans is our ability to get the right tissue at the right time, and that's a fundamental challenge. But I think if you were to look at cancer and ask what is the most surprising thing that has come out of genomics in cancer in the last 5 years, it certainly would be the recognition of the wide spectrum of mutations in genes whose products modify chromatin. And Bert made the first discovery of this with IDH1 mutations. And the interpretation of that, of course, really goes along with the general point that epigenetics is going to play a major role in many of these diseases, and I suspect that the mutations that we're finding are really the tip of the iceberg and raise the question as to whether there may be non-genetic alterations that change the epigenetic marks as well.
Okay, well, I would like to thank our panel and thank the audience, and now turn it over to the mayor.